Hi everyone, and welcome to this poster presentation on simulating single-cell RNA-seq data controlled by causal GRN. My name is Yazdan and I'm a student here at McGill in Iman's lab. And today we're going to talk about GroundGAN, which is the method we developed. So one of the challenges that GroundGAN tackles concerns the lack of gold standard ground truth when it comes to benchmarking GRN inference algorithms. And we propose using simulation for this task and to create ground truth GRN. So our goals with GroundGAN are to realistically simulate single-cell datasets at both transient state and steady state in a way where genes are causally expressed under the expression of their regulating transcription factors. And that is done by letting the user impose a GRN onto the generation process. In addition to that, we also wanted to preserve gene identity, pseudo-time ordering, and to learn the causal nonlinear TF gene dependence and other properties of single-cell datasets such as technical noise, biological noise, all of that with no user inputs and only implicit parameterization. So GroundGAN is a causal implicit generative model, and we'll talk about the doors that add opens in a second. And as input accepts a GRN and a single-cell dataset. First, we use a single-cell dataset to train a Wasserstein GAN on all genes and TFs. And we then isolate the generator of this first GAN and rename it to a causal controller and use that in a second GAN. So in the second GAN, the causal controller only generates TFs, and those TFs are used as input to another generator called the target generator. And this target generator generates target genes based on TFs and noise to introduce stochasticity. And this is where we impose the causal graph, the GRN. That is done by letting the target generator's connections follow the connections in the GRN. And every single gene will receive as input its regulating TFs. But that is not enough to introduce causality. So we also have a range of other auxiliary tasks and networks that help us further impose the GRN onto the network. So GroundGAN, we showed how it generates realistic single-cell datasets. As you can see in this testnet plot, there's no evidence of mode collapse. In fact, this is not an issue that we face, and it's very common in generative models. And across a variety of metrics and datasets, we perform better than the state-of-the-art simulators. As you can see over here, across different distance metrics, maximum mean discrepancy, median integration latency scores, and when an RF is tasked to distinguish between cells generated from GroundGAN and real datasets, it performs quite close to random with about 0.54 AUC score. But that is not enough for us to achieve our goals. We also want GroundGAN to causally enforce a GRN. So in this experiment, we infer a GRN using GRN boost 2 on real data. And for every single gene, we use half of the inferred edges by GRN boost 2 as positive control and impose those edges onto GroundGAN. The other half we keep and do not impose. And that creates our negative control. And if GroundGAN actually does enforce the GRN, we expect GRN boost 2 and other inference methods to be able to pick up the edges that were imposed, the ones in the positive control. And that's actually exactly what happens. The edges in the positive control are now assigned higher scores by GRN inference algorithms, and we now achieve a higher AUPR on GroundGAN generated data on those edges compared to real. And the edges that were not imposed in the negative control are now assigned lower scores than their scores in real data. So their AUPRC drops down close to random. Okay. So we talked about GroundGAN being a causal implicit generative model. And what that allows us to do is to sample from observational distribution and also from interventional distributions. And that allows us to perform in silico knockout experiments and perturbation experiments. So here we have this experiment where we knock out the top 3 differentially expressed TFs of each cell type. And what we see is that the cells corresponding to that cell type are now dispersed into their neighboring cell types while still being inside the distribution of a single cell dataset. And that allows us to perform a variety of knockout experiments and perturbation experiments. And coming back to our original goal of benchmarking inference methods, we benchmark a number of GRN inference methods, and Our results show that GRN-Boost2, Gini3, and PIDC perform best across both datasets that we tested on and across different kind of ground truth GRNs. And our results were consistent with Beeline's results on curated models and real experimental dataset. And here it's noteworthy to know that many of these methods that do require pseudotime perform quite well on simulated datasets, but their performance drops down to random when ran on curated models and on real experimental datasets. And when we use GroundGAN to benchmark, we actually obtain results that are consistent with Beeline's results on real datasets and not on simulated datasets. So we are closing the gap that exists between the performance of these methods on simulated datasets and real datasets. So we talked about some of these methods that do have pseudotime. So we also have a study that shows that GroundGAN can be used to generate continuous cell states, in which we show that we maintain the pseudotime ordering of cells, and that when we infer pseudotime using PAGA and diffusion pseudotime, we can match branches in the trajectory to real data, and the graph topology stays consistent. So that was a quick overview of GroundGAN and its applications, and thank you for coming to this presentation.